revision session of biology for grade 9 advanced end of term 1 exam. So this session we'll be covering the complete review for the session in 2 videos. Uh, Accordingly, we'll divide it into half. So this is the first video. So let's dive into it. And this is based on the coverage document which we received from the Ministry of Education. So if you see in this year, they have increased the number of questions. This year, the questions to be answered, multiple choice questions, will be 30. You have to answer 30 questions in 2 hours. And the lessons that are covered are mentioned here. So you have 3 modules, module 6, 7, and 19. Module 6, chemistry and biology, has 4 lessons. The first lesson will not be assessed, it is enrichment. Lesson 2, 3, and 4, chemical reactions, water and solutions, building blocks of life. Module 7 is on cellular structure and function. All the 4 lessons are included: cell discovery and theory, plasma membrane, cellular transport, and structures and organelles. Then the last module 19, introduction to plants, only lesson 2 will be assessed. And there is only page number 81 of this lesson from which the question would be taken. So let's dive into the review session. I hope you all are ready for it. So your first question, I have arranged, before I start, I have to tell you, I have arranged all the slides according to the questions, according to the lessons as they are in the book. So maybe you will see that this number does not tally with 1, 2, 3, 4, but the questions are arranged according to the lessons in the book. So we start with the first lesson, that is chemistry, chemical equations, chemical reactions. So the first question is talking about the outcome which is going to be asked is this, and it is on page number 11. Information from text means you need to go through the whole page and mark any important information that is there. So if it is information from text, please read the whole text, highlight it in your books, and form questions which could be asked. So page 11 is about reactants and products. Before you come to reactants and products, you should know what is a chemical reaction. So chemical reaction is a process in which the atoms, they are rearranging, the atoms or the group of atoms, they are rearranging and forming new substances. So some bonds are breaking and new bonds are being formed in a chemical reaction. So chemical bonds are broken and formed during a chemical reaction. For example, in this picture here, you can see this is a chemical reaction that is taking place. Rust is being formed. Rusting is a chemical reaction. A new substance, rust, is being formed here. So what's happening is that iron is reacting with the oxygen in the air. Iron is reacting with the oxygen in the air and is forming iron oxide. This iron oxide is what is called rust. And how do we know that a chemical reaction is taking place? There are some indications, some clues of a chemical reaction. Like they can produce, as a result of the reaction, there could be production of heat or light, and a new formation, new substance is being formed, new gas is being formed, or liquid or solid. They undergo changes and they undergo chemical changes. But in case you see this next picture here, water boiling. In water boiling, you can see bubbles coming and the gas is being formed. But this is not a new gas. It is water in the gaseous state. So this is a physical change. So you should know about physical change and chemical change. Then what is a chemical equation? Page information from text on page 12 also. So chemical equation is how the chemical reaction is being expressed. How are we showing the chemical reaction in terms of symbol and formulae? There's a formula of glucose, chemical formula of glucose, chemical formula for oxygen. So when we are showing it in the form of a like chemicals and symbols, in form of, this is known as a chemical equation. So when scientists write chemical reactions, they express each component of the reaction in a chemical equation. In written chemical equation, chemical formulas, they describe the substance, and the arrow describes that these substances are reacting to form what substance. The process that is taking place, the change that is taking place is shown by this arrow here. So the substances, we call them reactants and products. Reactants are those substances that are on the left side of the arrow. These are the reactants. And products are those that are on the right side of the arrow. These are the products. Reactants are the starting substances, and products are the substances that are being formed. Arrow means produces, yields, reacts to form. So here, like glucose and oxygen are the reactants, and carbon dioxide and water are the products. So glucose and oxygen are reacting to form. Arrow means to form carbon dioxide and water. So reactants and products. Now, whenever we are writing a chemical equation, we always make sure that the reaction is a balanced reaction, a balanced chemical equation. Why do we balance it? Because we know that in a chemical reaction, matter cannot be created or destroyed. It is not created or destroyed. Only it can change. So this principle, which is followed, is known as the law of conservation of mass. So all chemical equations must be balanced. This means that the number of atoms on the reactant side of each element should be equal to the number of atoms on the product side, or the mass of the reactants should be equal to mass of the products. So to balance it, like this one is an unbalanced equation. Because if you see here, there are 6 carbon atoms. Here, there is only 1 carbon atom. So it means it's unbalanced. So we'll have to balance it. Now, to balance it, we write numbers in front of the substance. It's written in front, before the substance. We do not write it in between. We cannot change the chemical formula. And we will write it in such a manner that that the atoms before the reaction, or the reactant, atoms of the reactants is equal to the atom of the product. So here, like there are 6 carbons, so we'll add a 6 in front of this carbon dioxide. So it becomes 6 carbon. But now when we write, this is known as a coefficient. This number which we write in front, it is known as the coefficient. And coefficient should be, like, we multiply it with all the subscripts. Subscripts are these numbers which are written here. These are called the subscripts. So we multiply the coefficient with all the subscripts to find out. Like 6, there is no number in front of C. So C means 1. No number means 1. So we multiply 6 by 1, it is 6. So there are 6 carbon. How many oxygens? We multiply 6 by 2. That means in this particular, there will be now 12 oxygens. Here, how many oxygens? Again, there is a 6 here. Multiply it with O. So it becomes 6O. So 12 oxygen this side and 6 oxygen this side. So total oxygen on the products will be 12 plus 6, 18. Now we'll check from this side. How many oxygens are here? Here we have 6 oxygens in this compound, and in this there are 6 times 2, that is 12. So 12 plus 6, 18. So we have 18 on one side and 18 on the other. Now it is balanced. So this is important to balance chemical equations, because according to law of conservation of mass, matter cannot be created or destroyed in a chemical reaction. So here they say in question number 2, they will ask you about reactants and products. You should know what are reactants. You know that on the left side are the reactants and on the right side are the products. So in this case, like CH4 plus 2O2, these are the reactants, and CO2 and H2O are the products. Reactants are the starting substance, and substances that are formed are called products. This is what will be asked from the first lesson. In the second lesson, you have to see the figure on page 13, figure 14. So here they are talking about activation energy. What is activation energy? So here you see, it says if you light a matchstick, you'll have to light a matchstick to start this reaction. So what are we doing when we are lighting a matchstick? We are providing some energy. We're giving some energy to the reaction to start. So this minimum amount of energy which is used to start a reaction, this minimum amount of energy is known as the activation energy. It is used to begin a reaction. So the flame of the match is providing the activation energy to start the reaction. Now, if you see this graph, here the energy and here is the reaction progress. So the reactants have this much energy and the products have this energy. So do you think that we can see in this graph that the energy of the reactants is more than the energy of the products? So what happened? Where did the energy go if the reactants had more and the products had less? It means the energy went out of the system. It was released. And when an energy is released, we call it an exothermic reaction. It exits, it goes out. Energy is released. How will you find what is the activation energy in a graph? So to find the activation energy, you may draw a horizontal line from the reactants to this graph, to the graph here, and you will see that this energy which is above this line, this is known as the activation energy. And in this case, this energy is the energy that is being released. So this is an exothermic reaction. Here it says summarize the meaning of activation energy. What is activation energy? It is the minimum energy needed for the reactants in a chemical reaction to form product or to start or to begin the reaction. Next is they're talking about enzymes. Information from text page number 16. Now, when we're talking about enzymes, the reactants, enzymes are catalysts, biological catalysts. What are enzymes? They're biological catalysts. They're found in living organisms and they help to speed up a biological reaction. So what happens? How do they do that? The reactants, they bind to the enzyme. And the reactants that are binding to the enzyme, we call them substrates. Singular substrate. So they are called substrates. And where do they bind? They bind at special locations, specific locations. Every enzyme has a specific location, so it can only bind with specific reactants, not with each and every reactant. So they are specific and they help in catalyzing the reaction. So the specific location where a substrate binds on the enzyme is known as the active site. The active site and the substrate, they have complementary shapes, just like puzzle pieces. They can fit into each other. So they have complementary shapes. And the structure of an enzyme is related to its function. So the substrates with the same size and shape as the active site will only bind. No one else can bind. Once they bind to the active site changes shape and forms the enzyme substrate complex. So once the enzyme is binding to it, the shape changes and it forms a complex which is known as the enzyme substrate complex. The enzyme substrate complex helps chemical bonds in the reactants to be broken and new bonds form. So when new bonds form, new substance form, then they are released. They react to form products and the products are released. The enzyme is then again available to bind with other substrates. So enzyme is not being used. It is just catalyzing, making it faster, and then like using more reactants too. They affect many processes. When a person is bitten by a venomous snake, the enzymes in the venom, they break down the membranes of that person's red blood cells. Hard green apples ripen because of the enzymes. Photosynthesis provides energy for the cell with the help of enzymes. So they are just like worker bees. They are important for the survival in the beehive. Enzymes are workers in the cell, chemical workers in the cell. This is an enzyme. If you see, this is the shape of the enzyme. It has a particular shape. So only those reactants which can fit into this shape like a puzzle, they can only bind with this. So the reactants are called substrates. They will bind and they will fit into this. Once they bind, it changes shape, and this is known as enzyme substrate complex. This is the enzyme substrate complex. Then the bonds are broken, new bonds are formed, and the new product is formed, which is released. And then the enzyme is ready to take another substrate. So substrates interact with enzymes at specific places called active sites. Only substrates with a specific shape can bind to the active site of an enzyme. Once the substrate binds, the active site changes shape and forms the enzyme substrate complex. The substrates react to form products. The products are then released. So this is about that. Then we come to the next question. It is information from text page number 18. So it's about water and solutions. And it's asking that this is the learning outcome. Solubility as the ability of the solute to dissolve in solvent. So first you should know what are intermolecular forces. You know that they are positive ions, negative ions, and they attract electrons based on their attraction, like ability or electronegativity. So the nucleus of an atom that has weak attraction will donate electrons to the stronger attraction. So similarly, atoms in covalent, this happens in ionic bonds, but in covalent bonds, they don't attract electrons equally. What's happening that some covalent compounds have atoms that hold on to electrons more tightly than other atoms in the compound. This results in a phenomenon that is known as polarity. So water is a polar molecule. It shows polarity. And this polarity is very important for water. It is a property of having two opposite poles or ends, like a positive pole and a negative pole. They have unequal distribution of charges. They are known as polar molecules, meaning that they have oppositely charged regions. Water is a polar molecule. It has slightly negative and two positive ends. Have a look at this figure here. This is oxygen atom. These are two hydrogen atom. So the electrons are pulled more towards the oxygen atom side because it's more electronegative. And so that's why oxygen side of the water becomes slightly negative. The hydrogen, since the electrons are being pulled towards the oxygen, it gets a slightly positive, and so it's a bent shape also. Water molecule has a bent shape, so that also makes it more like more easy to make a polar molecule, or that could be a reason for making a polar molecule. So slight negative and slight positive. And what is the reason for polarity? Electrons are more strongly attracted to the oxygen nucleus than to the hydrogen nucleus because it has more positive charge. So it is more strongly attracted to this nucleus, resulting in an unequal distribution of electrons. This along with the molecule's bent shape. So one is this reason, the other the shape. Creates oppositely charged regions. What happens? What is the effect of this polarity? Because oppositely charged regions attract, water molecules tend to form hydrogen bonds with other polar molecules. So they forming bond with other polar molecules because they are slightly positive negative. So they form bonds with other polar molecules. And that is the reason why they can readily form solutions. Why do they form solutions? Because of polarity. So this is the same figure as I just explained to you. Water molecules have a bent shape and electrons are not shared equally between hydrogen and oxygen. This results in the molecule with oppositely charged regions. Now there is a property of water which helps some of the insects like this water strider to walk on the surface of water. How can it walk on the surface? Because of the attractive forces between water molecules. We just did that there are hydrogen bonds, attractive forces between water molecules, which helps the water strider to walk onto the water. These forces are called van der Waals forces in water. We give them a specific name. We call them van der Waals forces. Consider how van der Waals forces work in common substance, that is water. You know that it's a polar molecule. It has slight positive and negative charges around the water molecule. They are attracted to the opposite charges of nearby water molecules. So that all the water molecules are being attracted towards each other and they're forming bonds. These forces hold the water molecules together. And because they are holding the water molecules together, it makes it easy for the water strider to walk on it. Without van der Waals forces, water molecules would not form droplets, and droplets would not form surface of water. The water strider takes advantage of this attraction. It is important to understand that van der Waals forces are attractive forces between the water molecules. So you have to remember that these are forces between water molecules, not between the atoms forming water molecule, like hydrogen. Okay, it's not between hydrogen and oxygen of the water molecule. It is between two or more water molecules. So when we're talking about this, page 21, figure 23, this will come as another question, number 15. Like here, here they're showing you the hydrogen bonding. So this is the water molecule, slightly positive, and this side is slightly negative oxygen side. This side is slightly positive. You can see this. Slightly negative, slightly positive. So the negative side will be attracted towards the positive sides of another molecule. So here you can see this is attracted towards a positive side of another water molecule. The negative side will be attracted to the positive side of another water. So this is how they're forming bonds. These are the hydrogen bonds. Now, because of these hydrogen bonds, it has specific properties. There are two important properties which you may be asked. One is adhesion and one is cohesion. Now, adhesion is what? Water is adhesive. Water is cohesive when we are talking about the process. So water is adhesive because it is forming hydrogen bonds, not uh, with molecules, not of only water. It is forming bonds with other substances, other surfaces. Like in the capillary action, it is forming bonds with this capillary glass tube. Here it is forming bonds with this paper. So it is forming bonds with other surfaces. Capillary action is a result of adhesion. It's why water can travel up the stems of plants. So you can be asked which property of water helps water to travel in the stems of the plants. Water has to go from the roots to the leaves. So which property is helping it to move from the roots to the leaves? So it is adhesion. Water travels up the paper and carries the dye. This is adhesion. So and try to understand which process is involved. Cohesion is this when the water strider is walking or droplets are being formed, dew on the grass, droplets. This is because of cohesion. So cohesive means the molecules are attracted to each other so attraction is between water molecules. Here attraction is with other molecules on other surface. Here it is between water molecules. The positive polarity of the hydrogen atom is attracted to the negative polarity of the oxygen, like here. This attraction creates surface tension. So you can be given the word surface tension. So because of this attraction between water molecules, there is surface tension. And this causes water to form droplets and allows insects and leaves to rest on the surface of water. They can rest, they can sit on the surface of water. They can rest because of this property, cohesion. Another property which helps a lot in the survival of other animals is that ice, when it is formed, it floats on water. Now, why is it that ice floats on water? When liquid water is cooling, when it cools, when it comes to 4 degrees Celsius, then it becomes more dense than ice. Density of water at 4 degrees Celsius is more than ice. So what happens? Ice is less dense than liquid at 4 degrees. So it will float on the top. This also helps in the movement of nutrients within the water. And because of this, ice is floating and the fish can survive because ice floats and they can continue living and functioning below the ice in the water that is beneath the ice. Then we come to distinguish between homogeneous mixtures and heterogeneous mixtures. So what are mixtures? Compounds are those which are formed by chemically combining of atoms. Now, mixtures are substances which are not chemically combined. They're just mixing together. Like you mix sugar and water, you mix sand and sugar. They're just mixing. They do not lose their properties. So the components are not losing their properties. They are keeping their properties. And this new product is called the mixture. It is a combination of two or more substances in which each substance retains or keeps its individual characteristics and property. It does not change. Like here, the drink mix, when you mix it with water, you will see that it is uniformly mixed together, but it does not lose its property. It will keep its properties. So this type of a mixture is known as homogeneous. So mixtures are two types: homogeneous mixtures and heterogeneous mixtures. So you should know the difference between homogeneous and heterogeneous. In homogeneous mixture, the composition, it has a uniform composition throughout. Throughout, it will have the same composition, uniform. We also give them another name. We call them solutions. So solution is the term used for, another term used for homogeneous mixtures. So here, now, when a substance is mixing with another substance, then the substance which is dissolving it, that substance is called the solvent. It, it is a substance in which another substance dissolves. That is a solvent. And the substance which is dissolved, that substance is known as the solute. So you should know the solute and the solvent. In a solution, there are two components, a solvent and a solute. A solvent is a substance in which another substance is dissolved. A solute is a substance that is dissolved. For example, sugar and water or salt and water. If you mix salt and water, so water is the solvent and salt is the solute because it is dissolving in water. In our mouth, we have saliva. This saliva is a solution of water, protein, salts, etc. Air is a solution of gases. It has a mixture of so many gases. So if you should know what is solvent and solute. Then we come to the second type of mixtures. These are heterogeneous mixtures. Heterogeneous mixtures in which the components are not uniformly distributed. They are distinct. You can see them. They're distinct. Like here in this, you can see the sand and the water. You can make out where is sand, where is water. This is a solution in which there is sugar, salt, and water. We cannot make out where is salt, where is water. So this is homogeneous and this is heterogeneous. Salad containing lettuce, croutons, and dressing, it's a heterogeneous mixture. You can see the components. They remain distinct and you can tell what they are individually. Here you can see these two are different, heterogeneous and homogeneous. Over time, what will happen if you leave it? Over time, they will settle down. And because they settle down over time, we call it a suspension. Suspension is when the particles are settling down at the bottom. Now, heterogeneous mixtures are two types, suspensions and colloids. Suspensions are those in which one of the components settles down. In colloid, it does not settle down. It just it remains in the solvent. The particles of colloid are smaller than the suspension. Example of colloid could be milk, blood, fog, smoke. All these are colloids. Okay. Here it says distinguish between solution and suspension. So you know the difference. This is solution and this is suspension. A solution is homogeneous mixture and a suspension is a heterogeneous mixture. Next we come to the pH. What is pH? Now this is scientists have devised a method to measure how acidic or how basic a solution is. Is it acidic? Is it basic? And how much acidic and how much basic? So how do they do it? They measure the hydrogen ions in the solution. It is a measure of the concentration of hydrogen ions. Concentration of hydrogen ions, and that is called the pH. So pure water is said to be neutral, neither acidic nor basic, and the pH is 7. So 7 is neutral. Now if you go down from 7 to 0, you are going towards the acidic. Acidic is increasing. And when you go from 7 upwards to 14, the basic nature is increasing. So pure water is neutral. It has a pH value of 7. Acidic solutions have abundance of hydrogen. Another thing is that acidic have hydrogen ions. They give hydrogen ions in solution, and basic give hydroxide ions in solution, OH minus ions in solution, H plus and OH minus. pH values are lower than 7 in case of acid. Basic solutions have more OH minus ions and have a pH value higher than 7. They will have H plus ions, but there will be more OH. Most cellular processes occur between 6.5, this is important, buffers. It occur between 6.5 and 7.5. So these are the processes, cellular processes take place here. This has to be maintained. So the mixtures which are maintaining this pH, they are called the buffers. And why do they do it? Because they do it to maintain homeostasis. They control the hydrogen levels. If you've ever had an upset stomach, like too much of acidity in the stomach, we take antacid. This antacid is a buffer. It will bring down the acidity to between 6.5 to 7.5. So buffers are mixtures that can react with acids or bases. They can react with both. And the main thing is they keep the pH within a particular range. In cells, it is within 6.5 to 7.5 pH range. So that's of this lesson. Next lesson is about the building blocks of life. So what are macromolecules? You have to go through page 26, this one, table 1. What are macromolecules? Carbon atoms can be joined to form carbon molecules. Similarly, most cells store small carbon compounds that serve as building blocks of large. So they are small compounds or small molecules which are making big molecules. So the large molecules are known as macromolecules. They are formed by joining of smaller molecules, smaller organic molecules. These large molecules, we also call them polymers. Polymers is another name given to the macromolecules. What are polymers? They are molecules that are formed from repeating units of identical or nearly identical compound called monomer. So monomers are small compounds and they are repeating. They're going on repeating to form bigger compounds which are called polymers. They're linked together by a series of covalent bonds. Biological macromolecules, they are organized into 4 categories. We have 4 categories of macromolecules, biological macromolecules, and we also call them the building blocks of living things. They are carbohydrates, lipids, proteins, and nucleic acid. Carbohydrates, lipids, proteins, and nucleic acids, this is where they are found. Functions important. Carbohydrates store energy and also provide structural support. Store energy, provide structural support. Lipids also store energy, but they provide barriers. They make barriers, The plasma membrane is made up of lipid. It is a barrier. It allows only some substances to enter. Both of them store energy, but lipids store energy for long term and carbohydrates for short term. Proteins functions, they transport substances, they speed reactions, they provide structural support, they control cell growth. Very important stuff, these macromolecules. Then the nucleic acids, they are the RNA and the DNA. They are stored and communicate genetic information. So you should know the functions of all these 4 macromolecules. This is the same thing. So proteins are made up of amino acids. Carbohydrates are made up of monosaccharides. Lipids are made up of fatty acids and glycerol. Nucleic acids are made up of nucleotide. These are the monomers. And these are some examples of carbohydrates, hemoglobin, enzymes, triglycerides. These are some examples. Here use an analogy to describe macromolecules. Analogy means related to something which you know from your daily life. How do macromolecules look like? So here they have given you some examples, like a bicycle chain. A bicycle chain is made up by the same part repeating many times, it's making a chain. A beaded jewelry, it has beads and it's repeating, and it's making that jewelry. Brick wall, the bricks are repeating and making a wall. So the same way macromolecules have monomers which are joining together to make a polymer. This is that. Next, they want you to know more about proteins. Information from text page number 29. What are proteins? Protein is a compound made up of small carbon compounds called amino acids. So monomer is amino acid. They are made up of amino acid. Amino acid is made up of what? Amino acid is made up of mainly, you remember the video I showed you in class? CHON, C-H-O-N, CHON. CHON is carbon, hydrogen, oxygen, and nitrogen. They're made up of these four, and sometimes they also have sulfur. All of the amino acids have the same general structure. This structure is very important. They ask you sometimes, what is this? Or what is this? So you should know this is the carboxyl group. So the carbon atom in the center, carbon has a valency of 4. It can form 4 single bonds. So one bond it forms with carboxyl group, COOH. One bond it forms with hydrogen. One bond it forms with amino group, NH2. And one bond it can have a side chain. It could be any other carbohydrate, like it can be any other carbohydrate. So variable side chain, but these two important. Amino acids have a central carbon atom. We call the carbon can form 4 covalent bonds. One of these bonds is with hydrogen. The other 3 bonds with amino group, that is NH2. This line here is showing the bond. This bond here. So it's not minus, okay? This is a bond with NH2, carboxyl group, and then another bond with COOH with this, and a variable group, that is a variable side chain. The variable group makes each amino acid different. So there are different types of amino acid, and they're different because of this. How many types of amino acids we have? 20 different amino acids, because there are 20 different variable groups. And proteins are made up of combination of 20 different amino acids. Several covalent bonds called peptide bonds join them. Now this is a special bond found in proteins, which is joining amino acid. We call it the peptide bond. So you can remember P for peptide, P for protein. So they are joining amino acids together to form proteins. A peptide bond forms between the amino group of one amino acid and the carboxyl group of another. Where is it formed? It is formed, this is the amino group, this is the amino group of one amino acid and the carboxyl group of the other. So it is a bond formed between carboxyl group of one and amino group of the other amino acid. I think that would be clear. What is the function of protein? 15% of our total body mass is involved in nearly every function of our body. Our muscle, skin, hair, all made up of protein. 10,000 different proteins our cells contain. They provide structural support, transport substances inside the cell, between the cells, communicate signals within the cell, between the cell. They speed up chemical reactions and they control cell growth. So please go through the proteins. And the same thing, figure 33, page number 29. I already explained you this. This is a general structure of amino acid. And then you should know about the peptide bonds. That's in this lesson. We come to the next lesson about cell theory. History of cell theory. This lesson you have done it as a cell study lesson. So please go through it again. Page number 37, all the content. Let's do it here. So it was like many centuries, scientists were not able to get to know more about the cells because they could not magnify the cell so much. But once the microscope was invented, then we got a chance to see the cell. So it was in 1665. Remember the date? Robert Hooke, remember the name? He was the English scientist who, with his simple microscope, he first time saw the cells in the slice of cork. Remember, he used cork to see the cell. Cork is found, it's dead cells found below the bark, okay, of the tree. So he saw in the oak bark. Sometimes they ask you, where did he see the cells? So it is in the cork cork of which tree? Of the oak tree. And when? In 1665. How? Through his own simple microscope. His own simple microscope was a basic light microscope. And he saw this is the structure which he saw base. He saw like, empty chambers. And this is a light microscope. It's magnified 100 times. He observed small box-shaped structures such as those shown here, and he called them cell. He gave the name cell because it resembled to small chambers of the monks in the monastery. So he gave the word cell. The term cell was also given by Robert Hooke. What is cell? It is a basic, this is the definition of cell. It is a basic structural and functional unit of all living organisms. Basic structural and functional unit of all living organisms. Then after that in 1600s, later, there was another scientist whose name was Anton von Leeuwenhoek. Another name you need to remember. Anton von Leeuwenhoek. He designed his own microscope and he then saw the cell again. But how was it different? He saw living organisms. He saw living cells. He saw living organisms in what? In pond water, in milk, and various other substances. So he saw the living cell. After that, many investigations took place and many more development of technology. And there we got to know more about the cell and different branches of science were formed. This is the figure, this is the structure which he saw Robert Hooke under his light microscope. Then information from text, page 40. Then we came, as technology improved, we were able to make more elaborate, more microscopes which could magnify more. And these were the electron microscopes. So as they began to study cells, scientists needed greater magnification to see the details of tiny parts. So in 1930s, like it came to 1930s, another name to remember, Ernst Ruska, he developed the electron. Who developed the electron microscope? Ernst Ruska, remember the name. And how is it different from the microscope which Robert Hooke had developed, or the light microscope, the one which you use in the labs? That is a light microscope. We use light to see the slides, the specimens. So instead of lenses, the electron microscope used magnets. So the main difference, light microscope has lenses. Electron microscope has magnets. Light microscope uses light. Electron microscope uses electrons, beam of electrons. So these magnets, they aim the beam of electrons on thin slices of cells. We have to make thin slices of cells. This type of electron microscope is called TEM, or transmission electron microscope, TEM. Because electrons are transmitted, they go through the specimen to a fluorescent screen. We call it TEM, transmission electron microscope. Thick parts of the specimen absorb more electron, thin parts absorb less. So it is forming a black and white shaded image of the specimen, and it's a two-dimensional image. So TEM is making a two-dimensional image, shaded black and white. Then, but it had advantages and disadvantages both. Advantages that it could magnify 500,000 times. We could see the specimen. That was like very good. Disadvantage was that we could not see living cell. It must be dead. The cell had to be dead to be observed. It has to be sliced very thin. You have to make a very thin slice to see it so that the electrons can pass through it. And it should be stained, or it should be colored or stained with heavy metal. Heavy metals are like gold, etc. Those who have a heavier mass. So it has to, these were the disadvantages. So let's just say, advantage was what? It could magnify 500,000 times more. Disadvantage, that it could only see dead cell. It could only be, like the specimen should only be a dead cell or dead. And it should be very thin slices, and it should be stained with heavy metals. Right? Many modifications have been made to the original electron microscopes over years. Then came the next microscope, which was the SEM. After TEM came SEM. SEM is scanning electron microscope. Now it scans the specimen only at the surface, over the surface. It's not passing through the specimen. It is only over the surface. So it is producing a three-dimensional image. One disadvantage of using SEM is also that it can be only used for non-living cells and tissues, only non-living cells. So you should know what is TEM, what is SEM, what are the advantages, what are the disadvantages, and how they both are different. Okay. Then after that came another third type of microscope, which is known as this STM. So TEM, SEM, and STM. STM is scanning tunneling electron microscope. Scanning tunneling electron microscope. It involves bringing the charge tip, a charge tip, metal charge tip is brought near the specimen. And when it is brought near the specimen, very close to it, the electrons, they go from the probe, they go onto the surface, they tunnel through. They tunnel through the gap between the specimen and the tip. And then the scientists were able to create a three-dimensional computer image. So it could be colored. Computer image of objects as small as atoms. So it could magnify it like too much and give a three-dimensional image. You can see here, the dark colors are like where there are depressions or cracks, and the lighter one are where they are raised portions. So the DNA could also be studied with this, magnified with the scanning tunneling. So what do we see? That in TEM and SEM, there was a disadvantage that it could be seen for, we could use dead specimen, but in STM, it can be used for live specimens. This was one good point of it. The scanning tunneling microscope provides images such as DNA molecule in which cracks and depressions appear darker and raised areas appear lighter. Here they have said, page number 40, compare and contrast a TEM and a SEM. TEM and SEM, they're similar. Both of them are using electrons. They are different because TEM producing black and white shaded image and SEM is producing also a two-dimensional image, but SEM produces a three-dimensional image. So your question number 19 will be from this lesson, and question number 6. So you're going to get two questions from this lesson, question number 6 and question number 19. Okay. Then question number 20 is also related to the cells. It is also from the same lesson, information from text, page 41, 42. So what are the basic cell types? You know the cell theory. According to cell theory, cells are the basic units of all living organisms. 3 points of cell theory. Cell is the basic unit of living organism. Then cell is the, all living things are made up of cells. And thirdly, cells arise from pre-existing cells. By observing your own body and the living things around you, might infer that cells must exist. They can be in different shapes and sizes. You also know that they are based, the shape and size depends on the function they are doing. But there is one special physical trait which is common, one physical trait that is common in all cells, and that is the presence of plasma membrane. Sometimes they ask you, that person went to Mars and found a cell, and how will he know that he has seen a cell? What is the trait which he should see? So he should see that if it has a plasma membrane, it means it's a cell. So all cells have a plasma membrane. So what does the plasma membrane do? It controls what is going in and what is leaving the cell. You will be studying the whole lesson on plasma membrane. Most cells have genetic material in some form that provide instruction for making substances. Cells also break down molecules. Now there can be two types, basic types of cells, prokaryotic and eukaryotic. Prokaryotic cells are the first cells. It's said that all eukaryotic have evolved from prokaryotic. So prokaryotic are the first cells. Eukaryotic generally are larger than the prokaryotic cells. When you compare them, this will be larger. Both have plasma membrane, but this has other structures inside which are called the organelles, but here there are no organelles. They have membrane bound structures which are called organelles, but here no. They carry on different functions in the cell. So eukaryotic cells, they have a nucleus. Main thing, they have a nucleus and other organelles, membrane bound. Nucleus is the central organelle. It has the genetic material, the DNA. Multicellular organisms made of eukaryotic cells are called eukaryotes. Those that have eukaryotic cell, we call them eukaryote. It's not only multicellular, they also have unicellular organism like yeast, amoeba. They are eukaryotic and they have the organs. They are eukaryotes. Prokaryotic cells are simpler than eukaryotic. They have without a nucleus, no membrane bound organelles. Most unicellular organisms are prokaryotic, like bacteria. They are called prokaryotes. So the scientists think that eukaryotes have, evolved from prokaryotes. Scientists continue to investigate that there are two types of cells, and they said that the eukaryotic cells have evolved from prokaryotic cells millions of years ago. What did they say? They said that it is because of the symbiont theory. Symbiotic relationship is where both the individuals are gaining from each other. They are like benefiting from each other. So the scientists believe that there was a prokaryote living inside another prokaryote, and they both were benefiting from each other, and this is how the eukaryotic cell was formed. So what is the difference between, question number 20 is also from this lesson, like, what is the difference between eukaryotic cells and prokaryotic cells? So common is that they both have plasma membrane and they both are found in unicellular organisms. But eukaryotic are also found in multicellular. They have membrane bound organelles and they have a nucleus. Prokaryotic don't have membrane bound organelles, no nucleus. Example, bacteria. Okay, so till here I will close this video now because it will become too long. So I want you to go through the whole video and try to understand the lessons which are covered in this. And in my next video, we'll continue from the next lesson, that is plasma membrane. And we will cover the questions from plasma membrane and other lessons after. So please go through this, and later on we will be also discussing the end of term questions of previous years. So I will close this video now and meet you in the next video.